Hey everyone, and welcome to this explainer. Today we're diving deep into the future of the farm, and honestly, we're unpacking the wild reality of animal genetics. We're going to look way beyond those exciting, flashy headlines of agricultural breakthroughs to examine the actual nuts and bolts mechanisms at play here. And maybe more importantly, we're going to talk about the really complex realities of what happens when we successfully and, well, sometimes unsuccessfully, try to rewrite nature's code. So, if you're curious about the hardcore science that's literally driving our future food supply, you are absolutely in the right place.
To kick things off, let's just think about this for a second. How is a modern farm like a high-tech genetics lab? We usually keep these two totally separate in our minds, right? Muddy boots on one side, white coats on the other. But the reality today is that the modern agricultural baseline is just completely, inextricably intertwined with advanced biotechnology. The techniques used to breed the next generation of livestock, they aren't some sci-fi future concepts waiting in the wings. They are the highly active, everyday toolkit that commercial breeders are using right now to fundamentally shape the genetics of our entire food system, and they're doing it at a massive scale.
All right, let's get into Section 1, the Biotech Breeding Toolbox.
Now, artificial insemination, or AI, isn't exactly breaking news, but we absolutely have to recognize it as the critical, foundational vector for literally all the modern biotech that follows. It provides the sheer scale required to distribute superior and eventually gene-edited genetics across entire global herds. Without this delivery mechanism, the most advanced laboratory breakthroughs in the world would just remain isolated little experiments. It's essentially the commercial highway that allows a single, highly engineered genetic improvement to optimize an entire population with just incredible efficiency.
And building on that delivery highway, breeders use a deeply interconnected genetic toolkit. These aren't just one-off tricks. It's like a giant puzzle where the pieces build on each other. First, we use DNA fingerprinting as a genetic blueprint to precisely identify the absolute best, most resilient traits in a given population. Then, researchers can literally isolate those specific traits through gene cloning and safely store them in these massive, complex gene libraries. It's this whole interconnected system that allows the industry to move from just passively observing good traits to actively capturing and manipulating them.
Okay, let's dive into this. The actual creation of recombinant DNA. The basic mechanics are to split the DNA chemically using enzymes, splice it to isolate the exact gene fragment you want, and then insert that new recombinant DNA. Simple enough on paper, right? But the immense biological complexity lies in getting that newly inserted DNA to actually express itself correctly in the target organism. It's one thing to just insert a gene fragment. It is an entirely different, massive challenge to ensure that the new sequence integrates seamlessly without totally disrupting the surrounding cellular functions or triggering some unwanted systemic reaction in the animal.
Which brings us to the massive leap in precision offered by modern gene editing. Tools like CRISPR-Cas9, TALEN, and ZFN represent an absolute game changer in how we handle DNA. If the older recombinant methods were kind of like using a molecular sledgehammer, well, these are molecular scalpels. They allow researchers to target a highly specific sequence and execute an incredibly precise deletion or insertion. It's this unprecedented accuracy that has completely accelerated the timeline for commercializing really complex genetic modifications.
Moving right along to Section 2, Designing Better Farm Animals.
And this brilliantly illustrates what we're talking about. We now have pigs that have been specifically engineered using CRISPR-Cas9 to be entirely resistant to porcine reproductive and respiratory syndrome, or PRRS. Now, to understand why this one specific genetic deletion is such a huge deal, you have to look at the economic toll. PRRS costs the global swine industry billions of dollars every single year, not to mention causing immense animal suffering. By precisely deleting the specific cellular receptor that the virus literally uses to enter the pig cells, researchers have created animals that are inherently protected from the disease from the moment they're born. That is a real game changer.
Over in the aquaculture industry, Aquabounty Technologies use these exact kinds of genetic tools to engineer salmon that reach market weight far faster and using significantly less feed than regular salmon. But this represented way more than just a successful lab experiment. It marked a massive, historic regulatory milestone. By officially securing approval for human consumption, it proved that this highly targeted biotechnology could actually successfully navigate the incredibly stringent regulatory pathways, making it all the way from the research tank directly to the commercial seafood market.
Let's look at another specific example in Section 3, the case of hornless cattle.
So traditionally, dairy cattle grow horns, and those horns have to be physically removed to protect the herd and the farm workers. As you can imagine, this dehorning process is super labor-intensive and it causes significant stress to the animal. Because of this, breeding a naturally polled or hornless, high-producing dairy cow has basically been the holy grail for the industry. By successfully using gene editing to insert the naturally occurring hornless trait directly into the genome of a top-tier dairy bull, scientists were finally able to ensure that this highly desired physical trait was cleanly passed right down to the next generation of calves.
By late 2019, this success was making major news. The BBC was reporting on genome-edited bulls passing the hornless gene to their calves, and it really felt like the industry had finally crossed this massive threshold into a clean, precise, new era of gene editing. Researchers had effectively solved a long-standing agricultural challenge, massively improving animal welfare and farm efficiency without any apparent negative side effects. The incredible promise of the technology was seemingly being fulfilled right before our eyes.
But that brings us to Section 4, when gene editing goes wrong.
Yeah, just as the success of those hornless calves was being widely celebrated, there was a massive record scratch. A major regulatory setback occurred. FDA regulators, who are using advanced, open-source bioinformatics tools to rigorously examine the genome sequences of these supposedly perfect cattle, stumbled upon a critical anomaly. And what they found hidden deep in the genetic data literally brought the much-anticipated commercial barnyard revolution to a grinding halt.
So the crucial point here is understanding the exact technical mechanism of this failure. The intended result? It worked perfectly. The calves were completely hornless. But regulators discovered that the bacterial plasmid, which was the actual delivery vehicle used during the editing process, had inadvertently integrated its own bacterial DNA directly into the calf's genome right alongside the targeted trait. Actually, it perfectly highlights the hidden off-target risks associated with even our most precise tools. We can totally engineer the desired physical outcome, sure, but we are still navigating the immense and frankly sometimes unpredictable complexities of how these molecular delivery systems interact with living, breathing genomes.
Which brings us to the ultimate question we really have to consider as we map out the future of agriculture. How do we balance this staggering, world-changing potential with the kind of biological unpredictability we just talked about? Is the incredible promise of disease-resistant pigs and highly efficient hornless cattle worth the inherent risk of unexpected genetic insertions? The molecular scalpels we have today are extraordinarily powerful, no doubt about it, but they clearly demand immense precision and unwavering oversight. Something to think about next time you hit the grocery store. Thanks so much for joining me to explore this complex, truly fascinating science.